I'm Aurore Dupin, and I'm a synthetic biologist. So when I'm explaining what synthetic biology is, I like to take the example of what synthetic chemistry was a couple hundred years ago. So scientists looked at the molecules that you find in living systems, and they asked themselves if they could synthesize those molecules from scratch. On their way to making more and more complex molecules, they understood better how molecules are structured, how they function, how atoms bond. And not only this, they also expanded the realm of what can be synthesized beyond what existed already in nature. And today, those synthetic chemicals are everywhere in your life. It's the aspirin that you take when you have a headache. It's the nylon that's in your clothes. We use it in our life everywhere. Synthetic biologists, we look at living systems and at their basic building blocks, cells, and we ask ourselves, can we make cells from the bottom up? Can we make things that have a metabolism like cells, or systems that carry genetic information like cells, or that have a structure like cells? And on our way to developing the systems, what can we understand about how cells are structured, how they function, how they are built? And can we expand the realm of what can be made with cells beyond what's already available in the living world? So the very first function that you want to mimic when you're making an artificial cell is making proteins out of DNA. That's the central dogma of biology. That's the basic functions of all cells. And we can synthesize DNA. In order to make proteins out of synthetic DNA, there's different approaches in artificial cells. The one that I use is employing cell lysate. So basically, you take a living cell and you extract out of it the machinery that you need to make proteins out of DNA. And with this, we can express natural protein and non-natural proteins. And my lab is focusing specifically on trying to look at artificial cell in a spatial resolved manner. What that means is basically we're imagining making artificial cell as if you would take a cell and you would break it open and spread it out on the surface. We use silicon chips that are the same material that are in microchips in your phone. And we are able, so the lab developed a method to be able to bind biomolecules to silicon chips. So we can bind DNA, we can bind proteins. So we take those silicon chips and we carve out little compartments that are about 100 micron in diameter. In there we come and deposit our synthetic DNA. And when we incubate it with cell lysate, we express proteins that then come and bind on the surface around the DNA. So what we're having here is an artificial cell with a kind of genotype-phenotype linkage localized. This method has been used to build complex molecular machinery, to do dynamic circuits. What I'm focusing on is I'm trying to mimic a very specific function of your immune system called antigen presentation. So the immune system is complex in the way that it's structured and works, but basically its basic function is to detect foreign elements that enter your body and destroy them. The way it can detect foreign elements is in part using antigen-presenting cells. So what those cells will do is they will take a foreign element, let's say a bacteria or a virus, and they will chop it up into smaller pieces that they will then present on the cell membrane for the immune system to detect. Those little chunks of the bacteria or the virus are called antigens. Antigens are anything that will produce an immune response in your body. So the antigen-presenting cells present the antigens, and then the immune system comes and checks if you have antibodies that can recognize this antigen. If you have an antibody that binds this antigen, you start an immune response. How do we mimic this function of antigen presenting? We take our artificial compartments and we deposit on the surface DNA that codes for an antigen. So for us, an antigen is a protein or a chunk of a protein. We incubate those compartments with cell lysate, and we express our antigen, bind it on the surface in the compartment. Our antigens are labeled with the fluorescent protein that's indicated in blue here. So whenever you see a blue fluorescent signal, that means the antigen is expressed. When the antigen is then presented on the surface of our chips, we can come with an antibody and recognize whether this antibody binds our antigen or not. We can do this in a multiplex fashion, so we build chips that have 384 of those single compartments, and each of them can express and display a different antigen. So we can display an array of antigens, express them. So you see a diverse antigen expression in blue here, and then we come with an antibody sample, and we check which antigen this antibody can recognize and which it can't. The antibody we're detecting with a secondary fluorescently labeled antibody, so that gives you a red fluorescent signal. And you can see some wells are bound, some wells are not bound. All right, as a first proof of concept, we looked at very simple antigens. We took two short peptide sequences, His and flag, and a bigger protein, N. N here stands for the nucleocapsid of the coronavirus, of SARS-CoV-2. We expressed them in our artificial cells, and we verified that we have expression. So what you're seeing here, each little circle is a compartment. The black dot in the center is the DNA. The DNA is so dense and compact on those systems that it blocks anything else from binding on the surface. And around it, you see the antigen in blue that's bound on our surfaces. We then come with the antibodies that are specific for each of those antigens, and we're able to see very good orthogonal and specific detection. So the anti-His antibody only binds the His antigen, the anti-N antibody only the N antigen. So from this, we can see that we're having good expression of the antigen, proper functional folding, and good detection by the antibody. All right, so that was the first proof of concept. Now we want to do what your immune cells are doing, what your antigen-presenting cells are doing. So we want to do this process of chopping up a larger protein into smaller chunks. We do this at the genetic level, right, because we deposit DNA. So we put the full nucleocapsid protein, and we also display antigens that are small fragments of the nucleocapsid, 100 amino acid long, and they scan the entire sequence. We then come with two antibodies that we know bind the full nucleocapsid, and we ask, do they also bind the smaller regions? And the answer is antibody dependent. So we see that one of the antibody we tested only binds the fully formed protein, whereas another antibody recognizes very strongly a subregion between amino acid 250 to 350. What does that tell us? That gives us critical information about the epitope of antigen-antibody binding. So antibodies don't bind an entire protein, they bind a subregion of a protein. And this epitope is difficult to determine. Typically what we would have to do is purify the antigen, co-crystallize it with the antibody, and check the structure in cryo-EM, crystallography. Here we're able to very quickly scan different regions and check where the epitope is contained in our system. So that was mimicking what antigen-presenting cells do. Next, we wanted to go beyond what your immune cells can do. So we went into looking at point mutations because we know that viruses mutate, and we know that when viruses mutate, they can evade your immune system. We've all experienced this with the COVID infection. So we focused on the one protein in the SARS-CoV-2 that mutates the most frequently. That's the spike protein, and specifically a subregion called RBD. We're able at the genetic level to come and present on our arrays the wild type, the different variants, alpha, beta, delta, omicron, as well as the single point mutations that are present in those mutants. And when we come and assess the binding of an antibody, we're getting an epitope reading with a precision of a single amino acid. So for example, you'll see that mutation K417N here deletes binding entirely of this antibody to the antigen. This point mutation is also present in beta and omicron, and those variants do not bind the antibody. So in a putative immune system, those mutations would basically evade your immune system and be able to go through without being detected. We also see that other point mutations increase binding, some are ineffective to binding, and we have a very precise resolution of how the antibody binds. All right, so we've made artificial cells that can present antigens in a multiplex format. We use this to do epitope characterization of antigen-antibody binding, and we can go down to a single amino acid resolution. To finish, I just want to thank the people who make this work possible. So my lab, the lab of Roy Barzif, and the funding agencies that support me, EMBO, the Feinberg Graduate School. Thank you for your attention.